Professor Cameron Simpson, take one.
I know, I'm like, oh gosh, when I hear that title, it seems weird. Yeah.
Hi there, how are you?
I'm well. How about you?
Tired, hot and grumpy.
Could have been worse if it was yesterday.
It was yesterday. Thank you for sparing your time.
Pleasure.
Now, you got involved in science.
Way back.
When and why?
You know, it's funny, as a child growing up, I was interested in being a pharmacist. And I did pharmacy work experience and went, oh, I'm not going to do that. I'm not going to stick a label on a bottle every day. I never knew there was hospital pharmacy at the time. And then I thought I'd be a mathematician. And I went to university and did a basic science degree. And very quickly decided I didn't like math that much, but I thought genetics was excellent. And so from there, you know, we had prac classes back in the day that would go for 6 hours at Monash. And I really, really enjoyed genetics. I did some microbiology as well, but it was really the genetics that captivated me and basically followed my career from there.
But that's not high school.
No.
That was university.
So that was university, but in high school...
there must have been some catalyst where you wasn't just your mum being a...
No, my dad was a farm... I can't even describe Dad. Actually, that sounds bad, doesn't it? No, I was the first person out of our entire family that actually went to university, actually. So we were never really looking at academic careers, if you like, back in the day. And so, what would I say? You're going to edit all this as it goes, aren't you?
Well,
right?
that's
so
fine, but you don't answer anything.
No, I'm just trying to think, because I don't think I ever imagined myself being a scientist when I was in high school. You're trying to find something that's interesting to you, right? And you're doing fairly standard subjects, which these days the children have so many choices at school. It's really amazing. So I was doing this to standard biology, chemistry, physics, maths. And most of the focus in year 12 was maths, chemistry, and physics. So it was the high-level sciences, traditional sort of subjects, but I was never really an arts student. So it probably wasn't really until I went to university that I got some sense of what science actually meant and what I really wanted to do. So I think I was curiosity-led, eldest child, always out on the farm helping my dad, asking a lot of questions, working with the animals. So I really got sort of that curiosity from those days, but it really wasn't until I went to university that I got some subject matter expertise and understanding of what really interested me.
What did you sign up for after high school? Yeah, when you went to
science degree. A
BSc.
Yep, just a science degree. Yep, and I started with maths. What did I do? Maths. It was very basic. Maths, biology, chemistry, psychology was the first year uni, and I very quickly dropped the maths, kept the psychology as a minor, and then specialised in genetics and microbiology. And then genetics third year was my major. And beyond that, though, surprisingly, my early days postgraduate were in plant science. So I worked for Australia's very first biotechnology plant science company called Florigene, where we were trying to make a blue rose. And
did you?
Made a version of blue, which is still around. We were sharing that recently. We have a group chat for everyone that worked in that company. At its heyday, there was probably 50 people. So it was a unique experience to be working for a biotech company, small company. Everyone was very engaged and it was exciting science, but I couldn't really see where my career path would go from there. And I guess that coming back to being on the farm, the sort of interest in animals and human biology really kind of bore out, and I decided to take a PhD working on marsupials. And that again was something quite different. The biological question was understanding milk and milk production, and whether or not one day we could have transgenic milk that would make people, or that for people that could not tolerate lactose. So it's not coming out the right way there.
So the lactose intolerant people would not suffer.
Yes, yeah. So that was sort of one of the original premises, but really it was basic biology. And I think as a thread through my career, basic biology, even though we have translational science now, the basic biology is fundamental. Understanding.
What's translational science?
Well, translational science being in a cancer institute is where you're actually trying to apply your science to a medical outcome. Really. So you've gone from understanding the biological question to applying it to a treatment or a therapy. But basic science underpins everything we do. And so back in those days, it was understanding the biological, the lactation pathway of the marsupials and how that might relate to the human lactation pathway and the signalling proteins that are in there. And it was a unique experience.
Did it relate to humans?
It did. Well, the remarkable thing about the wallaby is that it's born the size of your thumb and it lives in its pouch for 180 days. And in that window of time, it matures. So it gets fingernails, it gets hair, its eyes open. All that process has to require different milk. And once it's born, it's more related to a human baby at birth, and the nutritional requirements are more similar then. So it was really quite an interesting process to see these different proteins turn on and off that were required for its development.
And I love it when you also turn the proteins on and off, and it's like we've got a big switch in our head that I see you visually. Well, that one's finished, turn on the next one.
Yep.
Is that sort of right? Could you paraphrase that for me?
Well,
my voice can't appear in it, so it's got to be yours.
The very remarkable thing about the wallaby is that even though it's got the same hormonal environment, and we have our babies and we have the natural hormones that come and stimulate milk production, the wallaby has 4 different teats, and the baby climbs in and picks one, and that's the one that grows, and that's the one that provides the milk. But you could reactivate there's a dormant blastocyst in the animal once they've had one pregnancy. They remate within 24 hours, and that remains dormant until there's a point where you may need to have that baby. So if the current baby dies, you'll reactivate it. Sorry, I'm not explaining this very well. I don't know how you can ever use this.
But you're saying you've got a baby on hold.
You basically do.
Okay.
24 hours after that baby's born, they will remate, and it'll grow to 100 cell stage, and then it will just be dormant. So if that baby is killed, for example, in the wild, they'll rebirth pretty much straight away. So the process of that is it's maintaining a hormone for having a blastocyst in utero in suspension. It's maintaining a baby that it's feeding. That same hormonal milieu should be regulating the mammary glands, and you can actually have discordant mammary gland milk and the same mother. It's fascinating. It's a fascinating area. Yeah,
it really is.
Yeah.
So after marsupials, that led you to? So
because I was working in the field of lactation, I decided to move into breast cancer, and heard whispers about cancer as I grew up, and some women that had succumbed to cancer, but it was never really talked about very much when we were children. But I always really knew it was there, and I thought, well, how do I take the biological knowledge I've got and apply it to something that's more high value, if you like, than understanding how lactation works in marsupials. There's really not an end game for that. It's interesting knowledge to have, but it doesn't really help anybody to know that. So I decided to work on breast cancer, and at the time, very eminent scientists Jane Visvader and Jeff Lindeman had just returned from Boston to start their lab at Weehai, and they didn't have a role for me at that point, but fortunately a colleague of theirs, Melissa Brown, joined the University of Melbourne, and I joined her lab to work on BRCA1, the breast cancer tumour suppressor gene. But we worked in collaboration with Jane and Jeff to understand a little more about the signalling of BRCA1 and to try and identify stem cells that drive breast cancer development.
So BRCA1's a place?
BRCA1 is a gene, tumour suppressor gene.
Can you better explain that?
Yeah. Let's see, when I started this postdoc, it was back in 1999, and the breast cancer tumour suppressor gene BRCA1 had not been identified for that long, maybe 3 or 4 years. Melissa Brown was an expert in this. She'd come from the ICRF in London, Institute of Cancer Research. What is it, ICRF? Now I've messed the story up. No. The ICRF, Internet
Research Cancer Foundation.
Doesn't matter. I just call it the ICRF. It was. Can you look it up, Danny? ICRF.
ICRF.
Yeah, I can start that again. Melissa Brown. Well, I'll just say the ICRF. It doesn't matter.
But you got straight from marsupials to cancer. You didn't.
I did.
Just straight to cancer.
Straight to that. So basically, the tenet here was the experiment and knowledge that I gained working in the marsupial model was able to be directly applied to working in a breast cancer setting. so that we could take human mammary gland material, we could culture it, grow it, manipulate it the way I'd been manipulating wallaby material and actually pigs in my PhD, and apply that in a human setting. So I had those skills that they didn't have. They were specialists in stem cells and in breast cancer and BRCA1. And so sort of married those two skill sets up for me to come and join that group and study tumour progression.
Makes sense.
Yes.
It does make sense. It
did.
How are we going to use this? You just explained how we
It did. It did make sense. It was a natural progression. I mean, the jump from plants to animals was career-defining change, really, because I really couldn't see where a career would go in plant science. And one of the exciting things about going and doing the marsupial work was that it included a post overseas for a year. And I thought, yes, why not? I haven't travelled before. I've never left a country. Let's go and see how it is somewhere else. And that was a really exciting opportunity. And you realise that science offers you those opportunities for the rest of your career. Conferences overseas, you make colleagues at international institutes, broadly collaborating with people. That's part of the real excitement of being a scientist. It's a global endeavour.
Did you find any scientific differences between the countries? I mean, I know, but you haven't said it was Massachusetts you went to. Wasn't it?
So when I first went to Scotland, I thought, I'm going to the Hannah Research Institute. This place is going to be amazing. I get there and it's a farmhouse in the middle of silage paddocks, and I was living on site there, and it was a good 20 minutes walk from the town, and the town was small. I thought, wow, you think the grass might be greener, but maybe it's not so greener. It was a great experience, though, to work there. But that fueled my desire to travel a bit more. And so the idea became, do your PhD, have some postdoc experience here, and then move on again. We had a two-body problem. My partner was also a scientist, or still is a scientist, and he was finishing his PhD and just wrapping up his papers. So, we had to synchronise that to be able to get jobs where we wanted to go. And after the experience of living in the UK, I thought I'd experience the US instead. And so we shopped around a little in the US. Back in the days, there was no Skype, there was no easy communication tools. It was emailing people, maybe a telephone call if you were lucky. So we sort of went cold. We targeted a number of places, and we did a little road trip. And students still do that these days. Postdocs looking for work will still go to a road show, visit labs, but it's so much easier to do now than it was back then.
The gypsies hunting for jobs.
Pretty much, yes. And when you've got two of you, that becomes a bit more of a challenge. So we went to DC, we went to San Diego, we went to a number of different places in the US, and I thought I'd landed a job, and it fell through. So we both looked at each other and went, let's just go to Boston. We know it's going to cost an absolute fortune, but what better place could we go to do our science? And I have to say it was the most wonderful experience to work there.
Because there's hardly any universities around Boston, are there?
No, that's right. You get to the Longwood Medical area and then you go, oh, there's the Children's Hospital, there's the Joslin, there's Harvard Medical School, there is everything. And at the beginning, there were seminars from every eminent scientist you could imagine any day of the week. And I started thinking, I haven't got time for this. I can't go and see these famous people because I actually have to do some work. But what an experience it was.
It was inspirational?
It really was. Yeah, and so much money. Back in those days, this was pre-GFC. It was, money wasn't a problem.
It was
ever.
buzzing.
Yeah, absolutely. Yeah.
So then what? The buzz came to an end.
No, I had a good stint there. We were in Boston for 6.5 years. So we were getting to the point where do we stay, do we go? I think there's always a strong family reason to come home. And we had one child there, and we thought time was probably right to return. So we basically weighed up what did we want to do, and that was my career defining point, where I decided that rather than head a traditional research group, I wanted to be a platform technology specialist and run a core facility.
You know my next question, don't you?
What does that mean?
Platform?
Yeah. So
it's going to be an important distinction.
So it is. Yeah. It is actually a really important distinction. I let Peter decide whether we were going to work in the US or whether we were going to come back here, and he found a position at Monash, and I said, right, that's it. I'm going to get a platform job. So I emailed Ricky Johnston and Grant MacArthur, who are both still here at Peter Mac. This is early 2008. and said, we're looking to return to Australia. I would love to head up a platform that enables people to do research in a specific technology domain, and whereby we work with them to provide them their experimental outcomes. And they were already very keen on the technology that I had been already well versed in the US. And we had a couple of telecons. We came out and I interviewed here. And I remember someone saying to me, I don't understand why you want to do this job. You know, you've had a very successful postdoctoral career. Why don't you go and run a traditional research team? I said, because I don't think there's room for everybody at the top. And I might be better doing something different than competing against all of my peers that I left only 7 years ago. And then I felt like I had more to offer the research community than being another person working in breast cancer.
See, you're confusing me with someone who understands what a specific technological domain is.
Yes. Right. So I wasn't sure how to bring that in.
Basic.
Yep. Yep.
I sort of understand, but I need you to explain it.
So which bit do you want me to explain? So
how it works, because you're sort of a provider, aren't you?
Yeah.
For different areas of science.
Yep.
And so what do you provide?
Yes.
That's a good start.
Yeah, okay. So the premise of setting this lab up was high throughput screening. So screening's a broad term. We started with the idea that we would be screening genes. So understanding the consequences of every gene in the genome in a particular biological context. So the method and technology we were using back in the day was RNA interference. And that's been surpassed by CRISPR technology now in probably the last, let's say, nearly 10 years. We've been doing CRISPR. One is a suppression of gene expression. The other is knocking out a gene. And there's other more complicated elements of CRISPR that can also suppress expression, but that was the distinction in the day. In fact, there was the Nobel Prize for RNA interference in 2006, and it was awarded very early on with the idea that it was going to be the game changer for everyone to understand gene function. So the whole lab was built on the premise that we could enable people to study every gene in the genome using robotics, using imaging, like you can see here, and using high-level data analytics tools to triage the list from every gene down to those that have very specific phenotypes that we can then go and study in the lab.
And the end usage of the importance of that is?
Oh, how do I describe that? I guess what's probably important to say is it's functional genomics. So I guess what I should say is all of that is, the terminology for all of that is functional genomics. Probably before the term genomics took on a broader meaning to be around sequencing. The idea of functional genomics back in the early 2000s was to understand gene function. So the broader context of that is that once you understand what a gene is doing, you can understand the pathway that it's impacting, you can understand ways that you may make mechanistic changes in your cells, or you may treat in a therapy manner.
That was good. I understood that. That was fantastic. Thank you.
Okay. I don't know how you're going to word edit out all my...
No, certain areas that we're travelling through. So it's not all a linear journey.
Yeah.
So, you decided to go down that multi-platform.
Yes. Yeah.
And then what? How do you start that?
Yeah. So, I guess because I'd been working in Boston, I was working with a screening facility at Harvard Medical School called the ICCB. Very famous facility for drug screening. They'd never done any RNAi screening before, so we worked out the technologies, the techniques to do that. And from there, I had a fairly strong background in using robotics. So from the training I had while I was in Boston, it enabled me to say, well, I know we know how to make robot protocols, we know how to shift liquid and do all of our cell experiments in high throughput. If we buy all the gear, we'll be able to do that in Australia and replicate that. And very few sites had that in Australia at the time. I was probably the only one that had that type of automation capability in Australia when we came back. And so, as it turns out, that year when I had negotiated to return to Australia, we had an application sitting in with the Australian Cancer Research Foundation. And I think I arrived in Australia inside of the 1 week of interviewing with the ACRF this grant bid that we had, and I was actually also pregnant at the time, and no one could know that. And so it was quite the challenge to sit there and be interviewed for this, and actually knowing that it was a fairly career-defining moment. If we didn't get the funding to set the lab up the way I had imagined it, what would I actually have come back to Australia for? So it was quite a lot riding on that grant application. And the ACRF do an interview every year when you're shortlisted. So we had the full day of interview and we pitched our vision. And a couple of weeks later, we found out that we'd got the funding. And so that's now 16 years ago. And that basically started the whole laboratory. If it wasn't for that funding opportunity, we wouldn't be where we are today.
Did you think it was make or break at the time? Or
I
do you
did.
think it was just something, I'll try for this and see what happens?
No, it was make or break. It was make or break. I don't know what the break would have meant if we didn't get it. I don't know what I would have done necessarily. I'm sure we would have made a go at it, but it was really, for me, absolutely essential. We got that money. We got $2.5 million, which in today's money is a lot. That bought us a huge amount of infrastructure. It bought us one of everything we needed to start the lab. And over the years, we've now up to 2, 3, and 4 of some of these items to enable our capacity and redundancy. But it was sufficient for us to have everything we needed to begin.
Okay, I'm going to ask one or two more questions, pass you over to Ben.
Yep.
But, you're going through cancer research, and it's all sorts of cancer research. This is where I get, because lots of different areas are coming to you.
Yes.
And I'm trying to explain what you do and how, and one, why you love your job.
Yes. Yeah.
And two, what it achieves and what you hope it to achieve in the future.
Okay, let me think about how to say that. So what I haven't said is what the name of the lab is.
That will probably be helpful.
So probably should say that, right? So our lab is called the Victorian Centre for Functional Genomics, but we are actually a national platform. So we have, in addition to the original ACRF funding and then subsequent opportunities where we've increased our infrastructure, we have funding from the National Collaborative Research Infrastructure Strategy, which is the federal government funding.
Again,
yeah, I probably need to, don't I? How do you edit? Can you just edit that bit? Yeah, okay. So we have funding from the National Collaborative Research Infrastructure Strategy, which is the federal government funding, and our partner is Phenomics Australia. And we also do work with Therapeutics Innovation Australia and Bioplatforms Australia that are all NCRIS funded bodies. NCRIS enables us to have salary support and some infrastructure support to keep the doors open. So how does that sound? Does that sound all right? So that gives us a little bit of a view into that. Got them all in. So it's important to list all three of them. So then what was the next question?
What do you love about your work?
Well, I guess what you were asking me before that, you asked me about the different science that comes in. So I guess what I can say is these days we're not just an RNA interference screening laboratory. We, of course, do CRISPR screening, but we also do a lot of drug discovery and drug screening. We're at the earlier stages of drug discovery where people might come in and set up their biological question and looking for targets that might be effective in their biology. That sounds shit. No.
You're allowed to change it. Okay.
Going to go back and reword that one.
Yeah.
But the idea of that to say we're not just RNA, I don't want to say we're drug discovery because what are we?
Well, in the simplest of terms, okay, just break it down as if I'm 3. What do you do? Okay, you take in cells.
Oh, thank you. I'm revealing myself.
How to see that?
It's got eyes. So I guess what I want to say is these days we're far more than just an RNAi screening facility. We do CRISPR screening, but we also do a lot of drug screening. And in that setting,
Well,
I'm just trying to think about what I want to say in less words.
What do you give towards cancer research? How about that? Hopefully, what's the best outcome for you and the company?
Yeah.
The laboratory.
Yeah. So I guess, for us, we support any researcher that comes into the lab from across the country. We're here to enable people to do their projects, and so the beauty of that is that we have our hands on all different biologies, and some really challenging and different types of biological questions come through to us, And it isn't just cancer, but we do a lot of cancer research. So probably one of the most exciting things we've done within the last year has been a personalised medicine drug screen, where the very talented PhD student, who is also a surgeon, would operate on patients with colorectal cancer. She would then take those cells that she extracted from that patient and make them into three-dimensional objects called organoids. We would then deposit those into a jelly-like matrix for them to grow, and we use our automation to do that. And then we would treat them with standard of care drug therapy that the patients could receive. And through some very sophisticated data analytics run by my team, we have been able to predict whether a patient will respond or not to treatment. So that's termed personalised medicine, and coming from the middle of this year, we'll be running a clinical trial based on this methodology that we've developed. So sometimes we're doing cell-based experiments that are very much the basic biology we've talked about earlier. And other times we've really been able to move into that personalised space where we are really able to make a difference using the tools that we have.
That was great. Thank you. I'm going to pass you over to Ben, the man with the black hat on.
All right.
Could you just cut that for a second?
Yeah.
You
okay?
Yes, I'm going.
Professor Canning, 1732.
We're all good.
All good, ready to go? Deep into each other's
So
eyes.
round two. I haven't done this before, so
I find that hard to
he's
believe.
done
Really?
this a lot.
Really?
Yes.
Why are you having a turn?
Well,
He's a writer.
because I need to learn.
Okay.
No,
Alright.
I wrote this.
Okay.
Well, I mean, you probably know all the background, but we're trying to get. Tom Derry wants a documentary done on cancer research, so that people give lots of money to the ACRF.
Yeah.
And
Yeah.
so that cancer is not conquered, but
Yeah.
managed.
Yeah.
And that led us to thinking, okay, the researchers need to be the heroes. And you kind of want to get to a point where grandparents are wanting their children to go and become cancer researchers because it's the coolest fucking thing. And what tends to happen, smart people end up being lawyers or bankers or, yes, doctors and that stuff, but then they might become an orthopaedic surgeon because that's where the money is, rather than actually becoming a cancer researcher. So I think what's happening that's great is you're discovering people who aren't kind of lab coat nerds. They're actually some real characters with some fascinating backgrounds. And yes, they're all intelligent, they're all passionate, but there's some really interesting things. And the other, we looked at a structure where we're exploring the history of medicine, then we're getting into what is cancer, and then the last bit is what are we doing about it.
Yeah.
And we've thought we've got these sort of things. Okay, first of all, prevent it is the best thing to do with cancer is not to get it in the first place. So we're talking to Ian Fraser about HPV development and stuff. The next bit is find it. And have we done the big skin cancer scan thing?
We haven't shot the big machine that goes down.
So which is
the smaller one.
all ACF things. And then there's
That's
the
in
blood,
Babylon, right?
the blood test thing that working with Johns Hopkins University, just so we'll be able to detect 9 or 16 cancers or something with the blood test.
Yeah.
Then diagnose it, identify it, detect. So working with ProCan, which what your last example sounded just similar to that. So you can identify what this is and suggest a treatment. Then we're looking at immunotherapy, and we spoke to Mark Shackleton just this morning.
Yeah.
We're looking at proton therapy, so a bit on radiology. We're not really chemo. We're talking to Griffith about developing drugs. So all these sort of things are going on, and I'm just telling. I'm giving you some background.
Okay.
So I'm just interested from listening to everything you said to Stu. You must have a unique perspective on cancer research in this country because you're collaborating with all these different institutions who are presumably all looking at things in slightly different ways. So, to that end, this is something we ask Mark. We're probably asking everyone. What's the future hold?
Yeah.
Like if you were putting money into cancer research.
Yes, you raise a really interesting question though, because every project that comes to us is unique, of course, and even if it's in the same disease model, different type of question. And so the biggest challenge for us is to find a model that best replicates the biology, and to help them miniaturize it so that we can do it in a high throughput way, we can get through it quickly, and so that it's statistically significant. So the basis of everything is a foundation model that's as close to biology as it can be, and a way to score for it. And the scoring for it and the analysis of that is probably what sets us aside from a lot of groups. In fact, because half of our team analyse data, and we're very good at innovating in that space and finding new tools and new ways to ask the questions and to tease out the data and get more defined information from it. But really for us, where we're heading, what's the biggest challenge is data analytics and compute, storage, speed. It's for us not necessarily the cell models, but actually how much data we can generate and how do we actually interrogate it.
So the biggest, if someone had $100 million for you, you'd spend it on a supercomputer or AI
we would.
Or something.
Yeah, we absolutely would. Yep. So I have to answer that, not So if we had $100 million, what would we do? We'd spend it on high performance compute, increase our speed, have more people doing the work, and spend more investment in AI.
So that would suggest, because the other thing, I failed a master's in applied biology because I didn't go to any lectures, but my methodology of my thesis was appalling. And I mean, I can remember in Oppenheimer, everyone said Oppenheimer's lousy at lab work. So clearly, great research depends on great thinking, but there has to be an element of being just the ability to design the models must be paramount.
Yeah. So I think one of our greatest skills and what we offer to people is that ability for us to hold their hand from the start to the finish. So they'll come to us and say, I've got this disease model, I've got a few molecules that I know about, I've got some drugs, here's my question. And we'll turn that into an optimized outcome for them to screen with. And so that's where our expertise really comes in the fore, because we've got that attention to detail. We know the statistical metrics that we need to meet. So we'll be setting up the most refined model system you can get so that biology can have a number. If we don't have a number on something, we really can't say it's better or worse than something we've done before. So the statistics behind it are really, really critical. But for us, the end user cares about their results. They care about their target hit list. We care about the method of getting there. So we will always look at screen QC. So quality control and making sure that every control that we've got on there, as robust as it can be, is paramount before we care about actually the outcome of any project.
So you can be certain of the result, whether it's a good result or not.
Absolutely. Yeah. If you don't have the right controls, you don't have enough of them, and they're not reproducible, you can't even start the experiment. That's really the foundation of all of it. So the really tricky experimental design require a lot of controls, just so that every time you do it, it's reproducible.
So I don't know whether it's possible to imagine we are PhD students and we've been working with Mark Shackleton, and we've found a pathway that there might be a switch in something to turn up the immune response, but we don't know what it is, but this is what it kind of looks like. How would you you take a brief, you basically take a brief, but then you develop a response with these. Could you give an example? I mean, it's probably
so if you came to me as a researcher and said, here's my biological question, I need to do a drug screen. I need to understand what compounds might kill my cells, for example. And we would ask you, what sort of cells do you have? What do you know about your cells? How do they grow? Should they grow as flat cells, cells in liquid, or in these three-dimensional organoid structures? Once we understand that, we ask whether there's any other things they know about their biology. Are there any specific markers that will give us an indication of changing in their cells after perturbation? So the student would then work with us to develop those cell assays, and we would spend all of our time hand in hand with them, watching them seed their cells, grow their cells, determine how fast they grow, and determine how we stain them so that we can get a readout on a microscope.
So do you sometimes have to send them back and say, can you do something? You're not there yet? Or is that confrontation with their
No,
prof?
in fact, that's probably the funniest thing of all of it, is that everyone says, book me in, I'm ready to go. We'll always say, no, you're not. There'll be quite a long road before you're actually ready to go. So, no, I think we try and set those expectations that optimization is the longest part of any experiment, really. And so we will have those to and fro's. And I can recall back in 2011, one of the very first whole genome RNAi screens we did was with the Australian Animal Health Laboratories in Geelong. And we literally had been working on this for a year. And I had the last meeting and said, if we don't try this one more thing, then I'll call it quits. We have not got enough robustness in our experiments to do that. So they went back, they tried again, and we hit success. And we ran that project, and there's now vaccine candidates for Hendra virus that we never had 5 years ago, that now is turning a lethal infection into something that can be treatable. So even though that's not cancer and it's human health, it's sort of underpinned. We were desperate to get this experiment to work, but we'd spent so long trying to get it to work that we had one last ditch thing to try. And once we'd tried that, we hit the method that we needed. So it is a little frustrating sometimes that optimization can take a long time, but I never like to use garbage in and garbage out, but we all know that saying, and we all know that that really is the reality. If you underpin so much expense and time on something that isn't as perfect as it can be, then you're wasting your time.
And do you...
No, I'm just...
Oh, what did I say? No, seriously, what did I say? Oh, yeah.
Leading
Yes.
Professor Canning Simpson, take your own.
Cutting edge of precision stuff.
Scientific research.
Yeah.
Um,
The question was about because you're in Peter Mac, you've got the ability to apply things
Sure.
to the
Yeah.
treatment.
Yeah. So
finish.
a lot of the work that we do is based on cell models, and I said earlier on basic research. But here at Peter Mac, we have the unique opportunity that we can engage with clinicians. And we have the unique opportunity. Oh, sorry, let me go back again. So, what do I say? So a lot of the work that we do is based on fundamental cell models and understanding disease mechanisms. But being at Peter Mac provides us with the unique opportunity to be able to directly impact patient precision medicine by being able to have access to patient material from people that are coming into the hospital, to being able to then transform that into a biological experiment that we can test in real time, so that we can actually turn around an outcome for a patient response within the period of time from which they've been being treated.
So what? So I didn't quite understand.
No,
so precision. What can you explain exactly what precision?
Yeah. So for us, we've been developing miniaturized cell models called organoids, which are primary material taken from a person that are cultured in their nascent cell model. So they've got other cell types mixed with them, and they form a three-dimensional structure. That three-dimensional structure is so much more reminiscent of the human body than it is when we do regular cell culture, where we've got cells that we've just been culturing over and over again. So the capability to interact with the clinicians within Peter Mac that are treating patients and are able to remove material from those patients, and for us to then be able to transfer those into a high throughput model, means we save on cells, we can test a lot more options, and we can have more replicates, which allows us to have more statistical significance and lost the train of thought there.
Maybe just maybe
Yeah.
start with what is standard of care versus precision medicine, and then how do you influence that?
Oh, yeah. No, but I just didn't finish that other sentence, did I? No, it's okay. I was just trying to think about. Oh, man. I'm not doing your best work anymore.
You're not doing badly.
Yeah, I'm just trying to think. No, it's just me. I'm trying to think about the things that we need to say. What am I trying to say? What was the question, Danny? You wanted to know about precision medicine?
I guess
Oh, standard of care, right. So, okay.
The majority of patients that come through will
Yep.
receive standard of care.
Yes.
There are a rare number of patients that they're not responding, and we need better solutions. How do we do that?
Yep, okay. So thank you. Going to say that again. So the way we've been applying our basic research tools, and in reality they're really quite sophisticated, the way we are able to grow ourselves in three-dimensional matrices, is that we can first evaluate how a patient responds to the standard of care treatment. And that is, person comes in with colorectal cancer and there may be a choice of 3 different drugs that they might be given that is based on the clinician's assessment and everything they know about that person's disease. We can test all 3 of those different treatments and work out which one most appropriately suits that patient. So when we can start to do that and have the high confidence that we're as accurate as we possibly can be and that our analytics tools are accurately predicting an outcome, we'll be able to say we can take your cells, we can culture them, we can treat them, and then we can tell you what you should be treated with. And that is the definition of precision medicine.
So it's like Goldilocks picking a bed.
Well, I mean,
that's probably far too. I mean, what you say, the 3 things are really interesting because you basically got these cells, because you can grow those cells really accurately.
Yes, yeah.
You can then be confident that the effect of the 3 different treatments is comparable.
Yep.
And then you recommend one of them. I'm
Yep.
just trying to find a parallel.
Yeah, so
Sorry, can you just brush that?
yeah, I thought that was annoying. Yeah.
It's not the porridge wasn't
Yeah.
Porridge.
Yeah, no, I mean, I guess the whole idea of precision medicine is our absolute accuracy. So it's predictive medicine as well. It's based on the more patients' data that you generate, the more accurate we can be. So we built our model on 1 patient, then we added another patient, and then we continue to add patients, and that gives us more confidence in our model to be able to predict an outcome. So it is really based on the fact that if we can kill those cells under that standard of care treatment, then we can then say to the clinician, yes, we would predict that this will be the drug that the patient should use.
And do you so you've got the 3 treatments that are established.
Yep.
Do you sometimes test a 4th compound with?
No, ethics doesn't allow you to do that. Yeah, so we actually,
If the patient allows it.
yeah, because we had done all this not on a clinical trial, so this was experimental only.
Right.
So in that setting, we have ethics that control what we're allowed to do with the tissue. And we did have one setting where there was a patient that was refractory to all treatment. And we thought, why don't we just throw something on? Of course, you can't throw something on. That's someone's life that's on the other end of that, right? So at some point, if you do drug discovery screens on these patient materials, then you can ask a much broader question. Is there some other type of drug that's going to give you that outcome? And those are the types of screens we do routinely, but they're not the type of screen that's ready to go into, saving someone's life. That doesn't sound very good. Does that sound all right? Like, is that...
Yeah, and maybe you can also frame it as it allows the clinicians to confidently go down one path where they might have had multiple options.
Yeah.
And you don't have to wait for the patient to try and then fail on one treatment before diverting to another.
So I guess if I say, what we're really hoping is that if we can get this type of tool off the ground, that'll allow us in the future to directly guide the clinician for the most accurate treatment that they may be able to provide. That sound all right? Sort of there.
Another crack.
Yeah, that wasn't very good, was it? Just trying to think. I mean, it's something that I'm nervous about saying because we're really only at the point where it's becoming possible. So to put too much gravitas on it right now where people go, wow, you can come to Peter Mac and you can take my cells out. I mean, I had a man a couple of years ago go, well, I've got money, can't you just do this for me? I'm like, oh gosh, there's so many hoops to go through for that. So I'm trying to be careful about not over-promising something. But I guess, really at the end of the day, the whole idea of being able to set this up now and test these hypotheses are that in the future, we hope that patients will be able to have their material extracted. We can culture them in these little mini formats, treat their cells with drugs, and hopefully then the clinician can come to a faster decision on the best course of treatment.
Yep.
That'd be better. Yeah.
Yep.
Yeah. Okay. We'll have to see how that goes. It is literally the best thing we have done in years. It is the most exciting thing that we've done, to be able to apply our basic research tools where we've set this method up, we've verified it, we've been doing it ad nauseum, we've built our analytics tools, to then be able to move into a true patient setting where we know one day this method may impact treatment decisions. It doesn't get more exciting than that.
What are analytics tools? I mean, I start glazing over thinking of statistics and I don't know, I've glazed over, but what?
No, that's a really good question. So the readouts that we primarily use are microscopy based. And in fact, I'm sitting right next to a microscope, but you wouldn't know that, would you? It's just a box. Highly sophisticated microscope is sitting inside. Our cells sit in plates that you can see right there, and they're stacked up, and they'll run all weekend, and we will come out with images that look like this. From that image, we've got to get a number. That might be how many cells there are, that are the dots. That might be how are the cells responding, which might be the red colour. And we need to score for changes in shape, changes in expression, so you can see bright patches and dim patches. So we will set up algorithms to find out as much as we can about those cells. Are they living? Are they dead? Then building on that, have they changed shape? Have they got other features that we actually can't necessarily see, but that we can computationally determine? And then when we've got all of that data in collection, then we will apply statistics to group them into functional categories. So then we'll group a whole group that have very bright stain, for example. And then we might group a whole group that are very dead, but very dead in a specific way. So once we can subclassify them, we can then work out which groups of molecules we want to work on further.
And do you...
Just before we do that again, could I just ask you to just make all the indications to the screen and to the plates and those other things?
Yep.
Just to do that really again.
So just to speak it, or just the same words again?
Same words, same, yeah.
Holy moly.
Maybe
Same ballpark.
Yeah, okay.
As succinctly as
Yeah, so what was I saying to you? You're asking me about statistics.
You
So
said, analysis.
Yep.
That box is actually
Yep, yep.
amazing.
So most of the work that we do is underpinned by imaging, microscopy, and you wouldn't believe it, but this is actually a microscope. You'd expect one that you look down with your eyes. Microscope in a box, a highly sophisticated unit. It runs 24/7. We will stack our plates up, and our cells are sitting in our plates, and it will just do automated imaging, well after well after well. And every one of those little plates has 384 single experiments in it. So there's a huge amount of questions we can ask in there. But then once we've got our images, we are looking at how many cells are there. These little dots represent a cell nucleus. Then we might look at how has the shape of the cells changed. And you can see the red colour there is reflected in black and white here. We've got bright patches, we've got dim patches. We can look at shape change of the cells, and that tells us something about their biology. So once we've gone through all of those plates, we've got a huge amount of data points. And from there, we use very sophisticated statistics to group the cell responses into categories that look the same as each other. So they might be very, very alive, very dead groups, some that have got very bright staining here, for example. Once we've classified all those, we're then able to look at those that look very, very different from our controls and pick those that we want to study in further detail.
So these looking at the same thing over a different period of time.
Yep.
To see how it changes.
Yes, so we can do them. We can do these type of experiments in time course. So does the cell response change over time? We can do it as an endpoint. 3 days after we've drugged them, who's living, who's dying, what's happened to the cells, have they changed shape? You can ask this question many different ways, but the fundamental of the imaging is that we are throwing in the pot a lot of different parameters, things that we can see and things that we actually can't see that are just measured by maths. And by the time we crunch the numbers, we can distill that into very finite signatures to understand the cell response.
And will AI play a huge role in the comparing the images?
Yes. So, artificial intelligence, machine learning, there's a lot of different terminologies for it, but we are definitely using that. And, one easy example is to say that, let's just say we're looking for very bright patches of red here. How many other wells have bright patches of red? So we could create a signature based on something we knew, and then we can go into all of those plates that are stacked up right there and say, find us every well that looks exactly like this signature. So we can reverse engineer almost what we're looking for if we know the phenotype that we're after. The reverse is true, where we use machine learning to open up a whole database of study that we know nothing about and try and find things that look the same as each other.
So I might be going off piece.
Yeah.
There is something I think we talk about in the history of science about the power of questions.
Mm-hmm.
I mean, it does sound like you've got to ask the right questions of this to.
So I think
get
at the
right
heart of
answers.
doing this type of work, it's fundamentally about setting up the right biological question and the most robust way to ask that question. Sometimes we don't have a cell model that can accurately depict the biology, so we need a surrogate. 20 years ago, people would use the same type of cells to ask everything, but if you're asking a question about breast cancer and you're not using breast cancer cells, you can't expect to get the right answer. So these days, it's very much about the model must best reflect the biology, but what else did you ask me? What was the rest of that?
That was
that all right, though?
I think we answered it. I think it was about the importance of questions.
Yeah, I mean, I think what you're asking me about was, robust cell models underpin success every single time.
I think there was also the philosophy of asking the right question if you ask the wrong question.
Yes.
Sorry, can I
No.
just expand on
Yeah.
that? Because one of the
No.
most, I can't remember who it was, but he said,
Malcolm?
no, there are no bad results. Like this notion, whatever you do, you learn from.
Yeah, actually.
As a researcher, you
Yep.
can't say it was a bad result.
Yep.
You can't say I failed. That was it. He
Edison.
said, no, it was one of the blokes who shot in Sydney.
Oh, okay.
The grumpy old one that Tom
Thomas
didn't
Edison.
like.
Worried about.
But he was saying, well, there's no such thing as failure.
No, you're right.
I don't know whether that's relevant because you're more. The model seems to be more robust that you developed here. So failure is not.
You can still fail when you do these type of screens if you haven't asked a rigorous question, because you can group anything that looks the same as anything else, but if you need some sort of very specific marker to tell you that relates to your biology, and that your cells are giving you the outcome that you expect. But one of the challenges of doing these screens is that you will always miss out on something. So you have to draw a line in the sand somewhere for the next group that you work with, and then you do that again. And every time you do that, you accept you will miss a little bit of something. But like anything, the low-hanging fruit will always be there. The things that stand out the most striking will always rise to the top when you're doing any of these statistical metrics. So, it's the art of identifying your target to follow is one of the greater challenges of doing this type of work. That makes sense?
I like art.
Yeah.
Sounds like a nice.
Well, it actually is. So when people walk away from a project like this and they've run all of those plates and they've been doing this for weeks and they've got a massive scale screen and we've grouped everything in our little statistical fractions, they still have to make a decision about what they work on after this. And that is a big challenge. You're faced with a huge amount of data. There's a world of literature out there. there is a lot of databases that we can actually bring in. And again, using artificial intelligence, we can overlay a lot of public domain information. But from there, the real art is identifying the winner. What is the target that you're going to follow, take back to your home lab and actually follow for the next 5 to 10 years? You've got to be really careful about making that decision and knowing you backed a winner.
Do you do an annual report?
Not too many annual reports.
Well, it just sounds like you've got this unique perspective on what's working and what.
Yeah, I mean, every year we probably support around 170 to 200 projects. Different types of biology, different types of experimental questions, experimental designs. So, many different disease models. At the end of the day, we don't get to see a lot of what happens for years after that. So, this process is years long. So the discovery time, building the model, running the screen, identifying the best targets to take forward leads you to, many years of subsequent research.
So there will be another Hendra virus vaccine, but you'd never know when. But, the equivalent for cancer, you think? Sorry, that's
what do you mean by that?
Well, we asked someone. I remember we said, so how long's the champagne in the fridge for?
Right,
at least 10 years or something like that.
Yeah, I think for us, the champagne comes out when you finish the screen, because that's a milestone in its own right. And what happens after that really depends on what the research team do with their targets. There's so many other, we call them omics technologies. So we're functional genomics. There's proteomics, metabolomics, lipidomics. There's many omics that can build on your story than just what genes do something or what compounds do something. So yeah, for us, we're happy when the screen is good. It's done. They've got their high confidence target list to take away with them. But the champagne for that research team will be many years down the track. Yeah. We're publishing stories now of screens that we've done 7 years ago, something like that. This is big picture science that takes a long time to unravel.
Cool. I was just thinking, if this was footy, I'd be taking your tips.
So family goes to Richmond.
Grand final in 2035.
2035. Look, my nephew plays for Fremantle, so I have to go with Freo. Yeah.
Can you think?
We're about to film some of your infernal machines outside.
Yes.
Would you like to describe what they do before we do it, so we know what we're shooting?
Yeah. Okay. So I guess if we go into the main robot room.
But address it to them.
Oh. Okay. Oh yeah. Of course. Yeah. So in order for us to process all these plates that you can see here, before we even get to the imaging stage, we need to use automation to deliver the cells into the plate, to add the liquid solution that the cells will grow in, to add the drugs, for example, or the CRISPRs. And to do that, we use high throughput robotics. So it's not like a robot with hands that walks along like you imagine from a cartoon. These are a little bit more boring like this. It is actually a deck where we will put all of the plates, and we have automation tips that will allow us to move the liquid around to every plate. It's a little bit like a Tetris. We've got all plates on the deck. We are delivering different agents to every plate. They'll then be put in an incubator where they will be living for the next 3 to 7 days before we will then use other automation to add the fluorescent stains that you can see here.
And that kills them.
At this point, that's an endpoint, yes. So once we add fluorescence, the cells are fixed and there's nothing, they're not growing anymore. That's the end of the experiment. We do a lot of live cell work though, where we are just looking at them in black and white imaging, and we can look at them every day in black and white, and we can actually still analyze them in black and white format as well. The power of adding the fluorescence, though, is it allows us to get more structural definition of the cells, where we can actually look at very specific cell organelles and understand how they change in response to perturbation treatment.
It's quite beautiful too.
Could we just talk about areas of brightness and the red bits again and indicate there? Just mind us up, sorry.
Sorry.
What do you want me to say?
blue bits
Yeah.
and
You were actually
nucleus.
talking, before you were talking about there are areas of brightness.
Yep.
and-
Yep.
Just talk about different areas.
Okay, so what we're looking at right here is just one field out of probably about 20 in one little tiny well of a plate. And effectively what we first do is stain to identify the cell. And the cell is most easily identified by the nucleus. Nucleus usually is coloured blue. And you can see it here in a single channel, it just looks black and white. But those little dots are all the nucleus. Some are very bright, some are very dim. This one here is just divided. So you can actually get a whole lot of information about the cell just based on the nucleus. The next channel, the red channel, is staining for cytoskeletal filaments, a little bit like our muscles. The muscles that help the cell get bigger, get smaller, contract and move around.
The muscles of the cell again, just tell me those.
What did I say? Sorry, the next one is the
So
red.
you're talking about the cells like they're like muscles?
Yeah, oh, they are. So basically, the red stain is picking up what is easiest to be described as the cell sort of muscle. It's an actin filament, and it allows the cell to stretch, to move, and you can mark the cell boundaries when they meet with each other by this stain as well. So what you can see here is very bright regions where the cells have all joined up, and more dim regions where there's less of the actin at the cell boundaries. We use that as a discriminator of cell shape. It's a really good marker for a very basic understanding of how did the cells respond to treatment. So you can see here this rounded up cell is actually this cell here. It's just starting to divide in half and make itself two cells. So we can see it's very round. So all of its actin is bright and ready to be separated out. These ones here are far more spread out. They're responding differently. They're far more spread out. That's not very good. I can't.
Are they older?
No, it's just in any cell population, some cells will be dividing and some cells won't be dividing.
Are these cancer cells?
These are, I'm going to say, these are probably breast cancer cells. Yeah.
What are the red guys? What's the red?
The red is this actin muscle marker.
Like we talk about the actual.
Yeah.
We talk about the muscle marker.
Yeah, so one of the fundamental markers that we use that gives us an indication of cell state is actin. Actin is a muscle fibre and allows us to see how the cells change shape. So actin we stain as red. It's almost just a pseudocolour. We pick it up in the red channel. We can look at it in the black and white channel. So each of these is a single channel when we acquire it, and then at the end we bring it all together as one fluorescent merged image. So we can see where the nuclei are that are now blue and where the cell boundaries are and any shape changes that have come about through the cells growing and being treated can be identified through the actin channel. Does that make more sense?
No, it's great. I was getting a variety of shots while you're doing
Okay.
it.
Yeah.
So that all makes sense.
Yep.
You can cut
Okay. Yep.
it down really well. That's good. Should we cut
Much
that?
like this, can we use
I
this to
just
subtype
one
the
simple
cancer?
question. What's a clinician?
What's a clinician?
Trust me on this one.
Yes, can you explain?
I know it sounds.
I know.
What do I think is a clinician?
No.
Watching this may
Define.
not know what a clinician
Define a
is.
clinician. Someone who treats patients.
Yeah.
There you go.
Well, there are two types of doctors in here. They're
right.
researchers
Yes.
and clinicians.
Yes. Like I always say when I get on a plane, I'm not one of those doctors. Don't come calling for me when things go wrong. So yes, I mean for us, the clinician is someone who treats patients, and the rest of us are PhD doctors, philosophical doctors, research based.
And what if you're a doctor?
Yeah, they just get doctor. Yeah.
Isn't worth it.
So some of those PhD students are already doctors, and they don't get a second doctor. Yeah. They just get more letters after their name. That's it.
It'll be worth
Yes.
something.
Yeah.
And just
Could you ask if you've ever seen any breakthroughs?
Oh.
Well, yeah.
Well, I suppose my favourite one is the Hendra virus, and that's probably not the one we want to highlight because it's not cancer. Have I seen any
No, but
breakthroughs?
it's also research. It's research. We're not.
You mean personally, or
Yeah.
what have I seen?
The blue rose. I mean, it could have been. Yeah.
The blue rose. I mean, did Mark Shackleton talk about his stem cell career?
Yes.
He did.
He talked about growing breast tissue.
Yeah. See,
Yeah. He said
I
that was
worked
in
with him on
America.
that. So yeah.
Oh, wow.
Yeah.
Were you in the same lab in America?
No, yeah.
Oh,
I was working with him
Well, that's
before I went to the US, and then he went to the US. So
That was nice. Eh?
I won't speak about that one then.
Well,
I guess
well,
if you worked on it too.
yeah, I mean, I guess you can, the idea is that, science is collaborative. For me, that's what drives me. Well, no, I mean, if that's what drives me is actually the collaboration and deciding the opportunity to enable people to do the best research they can do. But it's also a continuum. So a breakthrough today is only one part of the story, because the next breakthrough could be next year, could be 3 or 4 years from now. And I guess in one way, breakthroughs don't just happen. They are the culmination of many people having their hands on an outcome over time. For us in the lab, in fact, the way we view it is we look at the 1%ers. What are the small little things that we can do to make incremental changes, whether it's in a workflow that we do or whether it's in the next experiment that we've never done before and we can add some value to what people do, whether it's in the way we do our data analysis or how we return the data to an end user. For us, I guess we're not hunting for the breakthrough. We're really looking to help people make it, but it's a long journey. Is that all right?
That was bloody superb.
Just use that bit. Yeah, because, we had at the Peter Mac retreat a couple of years ago, we had a whole session on what's the next big thing. And I refuse to imagine we can imagine it because it doesn't just arrive, it is part of...
Can you do that again?
Yeah. I don't want to say that though because...
We refuse. You were talking about what's the next big thing.
So yes, when you ask me what is the next big thing, in my mind, you can't really imagine what that's going to be. I believe it is something that comes with time, it comes with conversation, it comes from collaboration. You'll have an idea, you'll socialize that idea, a few other people might agree with that idea, you'll expand that idea, and you keep going, and then all of a sudden you go, hey, look at that, it's a breakthrough. It takes time to reach that idea. Maybe only Sheldon on the Big Bang Theory has the single-minded focus to get himself the breakthrough that he always imagined. I love that show. It's funny because sometimes breakthroughs are, if I use the word posthumous, it's not exactly what I mean, but realised later. When you have a Nobel Prize for something, it's a game changer, but it could be quite a long time down the track that you realise the importance of it, because what happens subsequent to that delivers that first decision. So, yeah.
Related to this, or related to what Mark said about he was certain, if he said what the next big breakthrough would be, he was pretty certain he'd be 99% wrong.
Yeah.
So is anyone doing anything weird that you like? Are you usually working with orthodox approaches, or is anyone? Because he was saying when immunotherapy in the 90s was this weird shit that very few people were doing.
Yeah.
What's the weird shit nowadays? And is anyone you're working with?
Yeah, so we're actually spending a lot of our time trying to make really complex models. And given that immunotherapy is a very hot area, we're trying to find ways for which we can grow our cells in the little 3-dimensional units and add immune cells and work out whether or not they are targeting the cells as effectively as they might target when they go into a person. So for us, it's quite difficult to create those little models and they take a lot of time. So we typically, no, that's not a good answer.
if you look at Ben, you know, what you said earlier, when the scientists are sheep, you're not the first person to say it today.
I don't want to say that though.
Spotted any sheepdogs lately?
Yeah.
There is a theme that's emerging. That, you follow them to get the money. You need to be orthodox, and you need to show that we're doing this because of this. And you quote whatever.
Yeah.
So inevitably, you end up doing what you're following a similar path.
Yeah.
And it's almost predictable because people want it to be predictable.
Yeah.
But the most interesting things come from when you go
you're
off
right.
piece.
Yeah.
So
So I suppose you're right. When we do our experiments, we've got a safety net. We've set up our most sophisticated assay that we possibly can. But on the side, we're working on something else, trying to get that next method up and running. One of our greatest challenges at the moment is targeting genes in these three-dimensional organoids so that we can actually understand gene function when the cells are in a higher order structure. That's nothing we're funded for, but it's what we play with on the side to try and have that ready for the next person that comes in that wants to work with us. So a lot of what we do is tool development in our lab, in fact, and it is trying to foresee what someone's going to need and getting a head start on that so that when they come in, we're halfway there, and then we just use their cell models to finish it off.
That's very interesting too. There's something in advertising we talked about called the power of what the fuck, where you basically, everything suggests you should do this, but what if we did this?
Yeah.
And that's kind of...
I think my favourite saying to lab is just humour me and do it, please. And I had a postdoc years ago, and she said, well, I know we won't get the answer we want. I said, but let's just see. Yeah. And actually recently we did that. We have a method, we can sequence every well of these big plates, and that's 384 little sequencing experiments in one plate. And we wanted to know whether the immune cells were targeting the tumour cells and whether or not we could actually get a signature out from that. Everyone said, no, we can't distinguish one cell type from the other, so how can we actually get an outcome? Like, just try it. Just actually suck it and see. And let's have a look at what we get out of it. And we could. We actually sat there and went, here's our tumour cells by themselves, here's their signature. Here's it with regular T cells and their signature, and here it is with CAR T cells, and the signature was completely different. And we were all blown away because we didn't think it was possible. But it's a game changer because now, even if it's crude, from a screening viewpoint, we can use it to triage targets and then go back in and work at them in great detail.
That's great.
It's a power of assumption, isn't it?
Yeah.
Oh yeah, I am like the naughty one who just wants to just do experiments to see what you get from it.
Try adding a bit of earth clicker and see what happens.
Yeah, that's right.
That was an accident.
Yeah, but if you go into it thinking you know what you're going to get out of it, there's no point doing it. Did you get that?
That could be
it.
On that note.
And that note, I think, introduce us to your
Okay, let's go into the lab.
fabulous